Hello students, good morning.
Good morning, ma'am.
Good morning. Let's wait for 2-3 minutes for others to join. If anybody has woke up, okay. After that we'll start, okay? Okay, let's begin. Thank you both of you to join the session. Rest of your friends will join soon, I hope. Okay. This is not a regular class. So yesterday I told you to study regarding RNA. So I expect that you have studied it. And we will discuss the facts regarding RNA quickly within 15-20 minutes. Okay. So tell me, start with RNA can be, what is the full form of RNA? You both can unmute yourself and directly talk. Okay. That will be faster than typing messages.
Ribonucleic acid.
Yes, ribonucleic acid. Why it is called ribonucleic acid? Why ribo is there? Yes, Bandana.
Second position OH group is
Yeah. In the second carbon, actually that is not called like that. In second carbon, there OH group is present, and that particular OHO oxygen is lost in case of your deoxyribose. Okay, so that is the basic difference between DNA RNA in the carbohydrate thing. Okay, then the next thing is that where the RNAs are found in cell.
Nucleolus, ma'am.
Where they're found, not formed. Where you in the cell, in which section you can find the RNA. That is in both in nucleus and cytoplasm. Okay. Vandana, are you there?
Yes, ma'am.
Yeah, please unmute yourself and answer because we three are only here. No, so we can discuss, have a quick discussion. Okay, then you can continue whatever morning schedule you are having. Okay,
Okay, ma'am.
okay. Please answer. And the first time, who reported that RNA can act as a genetic material and where they have reported it? Yes.
मैम नो आइडिया, याद नहीं आ रहा.
Okay. Lokesh, can you?
No, ma'am.
Okay. Where, especially anywhere, you, have you found that RNA is genetic material? In which organism RNA acts as genetic material? It is in case of
Viruses.
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 Yes, viruses. Then the first ever time, in case of your TMV, that is tobacco mosaic virus, there they have found that it can act. It will act as a what genetic material. Why? Because the TMB consists of only protein and RNA, so any of them has to act as a genetic material, and the scientist name is Frankel Cornet and Jared and Scram. Okay, you will find this in your pathology section, but since it was related to RNA, that's why I put it here. Okay, so next thing is that what is the structure of RNA? Like RNA will be single stranded or double stranded or what it will be?
Single
Single
stranded.
stranded.
Yeah, basically it is single stranded. Rarely it is found in case in double stranded form. Where it can be found in double stranded form? Again, that is in viruses. Okay, in whatever exceptionalities are present, mostly those things are found in case of your viruses. Can you tell name the virus which is having double stranded RNA as genetic material? No.
No, ma'am.
So, okay. So basically, RNA viruses means normally RNA viruses are means most of the plant viruses are RNA viruses, to tell you generally, and most of the animal viruses are DNA viruses, means DNA as their genetic material. Okay, so in case of your lettuce big vein virus and alpha keto virus, okay, in those viruses double stranded RNA is found as your genetic material. Okay, and what is the base? If you see the bases, that is ATGC, what is the base composition of RNA? What difference you will find from DNA in
Adenine,
case of your
guanine, uracil instead of thymine.
yeah, uracil can otherwise be called as in form of, mean sorry, thymine can be otherwise called as 5
2-methyl
methyl uracil. Yeah, so
Yes.
only there is a difference of that methyl group, isn't it? But that changes the many properties. Okay, next thing is that your RNA can be found in both cytoplasm and nucleus. In which means in what form it is found in nucleus? It is found in the form of rRNA. When it is synthesized, rRNA is synthesized inside your nucleus, isn't it? And all the RNAs are synthesized in nucleus and they are transported to cytoplasm later on. Okay, then okay, so does RNA has any structural function? Means it acts as structure of anything?
tRNA is one structure, ma'am.
No, no, no. Means every molecule has its own structure, but in anything they act as building block, or they will help to make some molecules later on. ऐसा कोई मॉलिक्यूल है मतलब जिसको बनाने में आरएनए की जरूरत पड़ती हो, ऐसा कोई मॉलिक्यूल? that is ribosome. Okay, in ribosome formation and constitution, we there is the requirement of rRNAs. Rest of the RNA molecules are not having any structural function. They are catalytic or some other functions they are having. Okay, and RNA is a part of your central dogma, isn't it?
Yes, ma'am.
Yes, ma'am.
Yeah, RNA is part of your central dogma. Can anyone tell me what is central dogma means? How the flow of central dogma is?
Ma'am,
Ma'am,
DNA
the
to
information
mRNA to
is
protein.
transferred from DNA to RNA and then protein.
Yeah, means how the encoded information in DNA that is ultimately getting expressed. That is called central dogma, and the way between your DNA to RNA means DNA will be will be converted to RNA, and that can be reverted back. Okay, from DNA we used to make RNA. That is normal process. What is the process called for which converts DNA to RNA?
Transcription.
Transcription.
Transcription. Opposite thing, if you want to form RNA to DNA, what is that called as?
Reverse transcription.
Reverse
Reverse
transcription.
transcription. And there is another
Feminism.
term is there for reverse transcription. Anybody knows what it is called? It is also called as taminism. Taminism. Okay. Why? Because it is named after the scientist who discovered it.
Temin
Tamin
and
and
Baltimore.
Baltimore. Yeah. So both of them have discovered this process of reverse transcription. That's why it is called as taminism. Okay. So the next thing is yeah. So basically, five classes of RNA molecules. There are several classes of RNA molecules to tell you, but basically five classes of RNA molecules we found. First one is rRNA, mRNA, tRNA. Apart from that, snRNA and miRNA are important molecules. Okay, so starting with all these things, uh, um, which all of this will be ending off in protein product or not? Can anyone tell me? Means all this mRNA, rRNA, tRNA, all this thing, everything will end up in protein product or not?
No, ma'am.
Then which one will end up in protein product?
mRNA.
Yeah. So out of all these forms of RNA, only mRNA will end up in protein product. Rest of the molecules have some other functions, some catalytic or some regulatory functions. Okay, and that's why we can otherwise in other way we can tell that tRNA, rRNA, snRNA, miRNA, all these molecules are not translated. Okay. Okay. The next thing is that yeah. Hmm. Ribonucleotides are otherwise also sometimes called as ribotides. Means there is another way to call them. Okay, and DNA we may call that RNA is present in all living cell. Whether that statement will be true or false? Yes. RNA is present in all living cells. Whether that statement will be true or false?
False.
Why?
Ma'am,
Any
because it is,
just guess, yeah.
yes, ma'am, because it is also present in virus that is called as non-living thing before infection.
Okay, but I'm talking about cells. No, here we have not means in the sentence what it is telling. RNA is present in all living cells. In all the
Yes,
living cells, whether RNA is present or not. That is the thing. So the statement is true. Why? Because there can be some. If we say that DNA is present in all living cells, that might not be true. Why? Because some viruses are there where DNA is not found. Only RNA and proteins are found. But RNA will be found in all living cells. RNA is found in all living cells. Why? Because DNA directly cannot express, whichever organism is having DNA, it cannot express itself directly. It has to go through the section of formation of RNA, then only it can form the protein. But and that is the second case. This is the first case where DNA is the genetic material. Even if DNA is not present, RNA can directly go for production of protein. So whether the DNA is present or not, RNA will be always and always there in case of your any cell. Okay. Okay. Next thing is, mm, mRNA was first reported by which scientist? Which scientist had reported mRNA?
Jacob Bonbona.
Yeah. And mRNA is also sometimes called as working copy of cell. Why? Because it carries all the information that are present in case of your DNA. Second, next thing is that if we compare the percentage, means amount between the mRNA, tRNA, rRNA, which one is high, found in most abundant form?
RRNA.
Yes. Next to that, we can
tRNA.
tRNA, and mRNA is found in least amount. Okay. And the mRNA of prokaryotes and mRNA of eukaryotes, is there any difference between them? Nature of mRNA?
Yes, ma'am.
Yeah.
Ma'am, in eukaryote it is 70 years, in prokaryote it is 80 years.
I'm talking about mRNA. What you told, that is
Ribosome.
rRNA constitution you are telling. You are correct, but that is rRNA. Yeah.
Monocystonic and polycystonic.
Yeah. Prokaryotic mRNA is mono or poly?
Polymer.
Okay. Prokaryotic mRNA is polycystonic, eukaryotic mRNA is monocystonic. Why they are called poly and monocystonic?
Polycystonic is synthesis of different types of proteins.
Yeah, means cystron means the functional unit of gene which can form peptide. Okay. So because prokaryotic mRNA is continuous and it is not having, means gene means the genome of prokaryote is not having intron sections. So basically one stretch of RNA can carry continuously many cystrons. That's why it is called as polycystonic. And from one mRNA we can form many polypeptides. But in case of eukaryotes, for one polypeptide, one mRNA will be there. That's why it is called as monocystonic. Okay. If we see the stability of the RNA molecules among which one will be more stable, which one will be less stable? Yes. What about RNA, mRNA? mRNA, whether it is more stable or not? Okay, so tell you the fact that in case of a prokaryote, the translation and transcription goes on simultaneously, isn't it?
Yes, ma'am.
Hello. Yeah. So because both the processes goes on simultaneously, that's why prokaryote RNA are very short lifespan. Means it only average lifespan is 2 minutes. Okay. After that, it will degrade. Means as soon as it is getting formed, it is getting translated. So it will degrade, no problem. In case of your eukaryote, its lifespan is some hours to some days. Okay. Okay. tRNAs are having good means what lifespan is quite good for them. Okay. The next thing is what is the full form of hnRNA?
Heteronucleotide.
Heterogeneous.
Heterogeneous RNA or heterogeneous nuclear RNA also we used to call it. So hnRNA is the premature form of mRNA. Okay. Why? Why it is called premature form?
Because it contains both non-coded and coded part.
Yes, because it contains both exons and introns. Okay. So it has to go through a process to form the mature RNA. What is the name of the process?
Splicing.
Splicing, yes. So the hnRNA has to go through a basic process of removal of exons, that is called splicing. Apart from splicing, it has to go through capping and tailing process also to form the mature mRNA. Now that the capping and tailing has come, so which molecule has added to form the capping?
Methyl guanosine triphosphate.
7, to tell you precisely, that is 7 methyl guanosine triphosphate. Okay, it is that methyl group is at 7th position, so it is called as 7 methyl guanosine triphosphate. And in which end capping is done, 5 prime or 3 prime?
5 prime.
5 prime. So and otherwise in tailing is done in case of your 3 prime end. Okay, so for tailing, what is the molecule that is added?
Poly A polymerase enzyme.
Poly A tail, that is polymer of adenylated residues. Okay, okay, so what is the function of 5 prime capping and 3 prime capping? 5 prime capping and 3 prime tailing? Yes, there actually means in cell exonuclease molecules are there. Exonuclease molecule means those molecules which can degrade the molecule from one side, means outside. If it is cutting from side, both the sides, that is called as exonuclease. Exo nuclease means degrading, nuclease means that can degrade the nucleic acid, that molecule is called as nuclease. If it is cutting from outside section, it is called as exonuclease. If it is cutting from somewhere inside, that is called endonuclease. So from protecting the RNA molecule from 5 prime exonuclease and 3 prime exonuclease activity in both the sides, this cap and tail is added, right? Now yeah, coming to the next molecule, that is rRNA. rRNA was reported by which scientist?
Kunj ma'am.
Kunj, yes. RNA reported by Kunj, and RNA is the most stable form of RNA actually. And where RNA is synthesized?
Nucleolus ma'am.
Sunandan, are you there?
Yes, ma'am, I'm there, but I'm busy, ma'am. Sorry.
You're busy. Okay, 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 okay. Okay. So rRNAs are most stable type of RNA, and they are synthesized in nucleolus. Okay. So there are several types of segments of rRNA are found in case of your prokaryote and eukaryote. So can you tell me what are the means in case of your 70S RNA, rRNA, that is found in case of your prokaryote, and 80S rRNA, that is found in case of your eukaryotes, right? So what is the constitution of 70S and 80S? With what building blocks it is formed?
30s and 50s ma'am.
Yes, 70S is formed with 30S and 50S.
In that 50s, 23 rRNA is there
Yes,
and 5s rRNA is there ma'am.
yes.
In
And
30s is
yeah, tell.
in 30s is 16s rRNA.
Yeah. There is some speciality regarding the 16S rRNA. 16S rRNA is very famous. Why? Because this 16S rRNA are used for phylogenetic classification. Your 6 kingdom classification was given by which scientist? 5 kingdom classification of your
Whittaker.
RH Whittaker.
5 kingdom was given by Whittaker. 6 kingdom classification, Carl Oz. Carl Oz has given the 6 kingdom classification, and there he has included another group, that is Archaea. Okay, so that classification he took the basis was 16S rRNA. The basis for 6 kingdom classification was 16S rRNA, and there that helped him to study the phylogenetic analysis, like which organism is actually ancestrally linked to which organism. Okay. Then rRNA sometimes have the enzymatic action. In that time, what it can be called as? Any idea?
No, ma'am.
Okay, so RNA is the molecule we used to normally call, like enzymes are proteinaceous in nature. Sometimes enzyme can be non-protein also. That is, the example is ribozyme. So RNA sometimes act as that catalytic function, that is act as enzyme. That form is called as ribozyme. You can see this particular function in in case of your peptide bond formation, in case of your proteins, like when translation is going on, new amino acids are getting coming to the ribosome, and peptide bond is getting formed between them. For that region, for that purpose, there is a enzyme required called peptide transferase. Okay, so that enzyme, there is no separate enzyme for that peptide transferase. This rRNA itself act as peptide transferase. It does the peptide transferase activity. So that time it is called as, it is used as a enzyme also. It has the enzymatic activity that time. Okay, there is some in case of your RNA, you would have seen that if this is the RNA, if this is the RNA molecule, from somewhere the start codon will be there, from somewhere stop codon will be there. What is these sections are called? That is before start codon, after and stop codon. Any idea?
Flanking region.
Not exactly. They are called as untranslated regions.
UTRs.
Yeah, that otherwise called as UTRs. UTRs, why, what they do means they are basically found in case of your mRNAs, and they are the sequences that is before start codon and after stop codon. Okay, and they are not exactly translated. Why? Because your translation will start from start codon and end with stop codon. So why this thing are extra present? What is the function of this particular UTR molecule is? Any idea what it will do?
No,
Stability.
ma'am.
Yeah, it provides the stability. It provides stability to RNA, and also it increases the translational efficiency. Okay, so that is about your rRNA. So next molecule is tRNA, right? And for tRNA, there is a famous model that is proposed for tRNA. What is that model regarding the structure of tRNA?
Clover mode.
Clover leaf model. Why? Because the clover leaf, clover, have you seen the clover plant? It has four leaves like this, and there will be stem like this. So your tRNA, it resembles something like this, like this, like this, like this. Fourth one is not there. This is the structure, means rough structure of your tRNA, right? Okay, so existence means who first time proposed the means saw or observed the tRNA, means who is the scientist?
Crick, ma'am.
Crick, yes. And there is another name for transfer RNA you can find, that is also called as soluble RNA. Okay, there is another name that is also called as soluble RNA or sRNA, right? Then what is the basic function they do? tRNA.
Transfer of amino acids.
Yes, they transfer the amino acid from wherever it is present to the what, your ribosome for protein synthesis. And okay, so who proposed this cloverleaf model? Cloverleaf model of
Robert
tRNA.
Hall.
Holly,
Robert
yeah,
Hall.
Holly. Holly has got Nobel Prize for that. I don't remember the year, but he got the Nobel Prize. And there is another three-dimensional structure that associated with your tRNA. That is, sometimes it is called as inverted L-shaped structure.
Kim and Clark.
Inverted L. Yeah. So who proposed that inverted L-shaped structure of tRNA? Do you know?
Kim and Clark.
Yeah. Good. Then, okay, let's talk about the structure of your tRNA. So, how many loops the tRNA is basically having?
3 loops.
Yeah, Vandana, try to participate.
Yes, ma'am.
Yeah. So, what are the three loops of RNA? tRNA, basically.
Thymine loop, anticodon loop, D loop.
Yeah. So, what is the function of that D loop?
Amyl acylation region,
Yeah,
recognition region.
yeah, means the amino acid synthetase, that enzyme will go and bind to that particular D loop. It is also called as DHU loop. Sometimes this is called like that. Second loop is your T size C, isn't it? In that T size C, what you can find, 7 unpaired bases you can find, basically, right? 7 unpaired bases you can find in that T size C loop. Then anticodon loop, why it is called anticodon loop? Why? Because it is having the anticodons of the, yeah,
Complementary sequence of mRNA.
yeah, the codons will be present on the mRNA, and the opposite complementary thing will be present in case of your anticodon loop. Okay, and there is another site, important site, that is present in case of your tRNA, that is amino acid attachment site. In which end, 5' end or 3' end, that amino acid attachment site will be present?
3'
Any
end.
idea? Yes, it will be present in case of your 3' end, basically. And apart from that, I talked about snRNA. So snRNA full form, anybody can tell? Yes.
Small nuclear
Small
ribonucleic.
nuclear RNA.
Small nuclear RNA, and that, what is the function of that snRNA? snRNA is part of
Ma'am,
your
that was involved
spliceosome.
in a mRNA.
Yeah. Tell.
Ma'am, it was involved in mRNA synthesis.
Yeah, it is involved in mRNA synthesis. To be very precise, it is involved in the formation of your spliceosome. Spliceosome is the molecule that will go and do the splicing activity. That it will cut, means it will go and bind to the mRNA, and it will cut the introns. Okay. Then you might not be knowing that much about miRNA, siRNA. That is not required that much also. Just you know the name. That is, what is miRNA full form? Anyone can tell? Yes. miRNA is otherwise called as microRNA. It is the small RNA, basically. It is very small RNA with only 22, 21 to 25 nucleotide with it. Okay. And they are involved in a very important function. They are involved in RNA silencing. Okay. The miRNA are involved in RNA silencing process. Okay. Then okay. Then what do you know about the RNA world hypothesis? What's that? Anyone? Any idea? What is RNA world hypothesis?
No, ma'am.
Okay. So RNA world hypothesis, actually it was proposed by Walter Gilbert, and what is said that means uh, we are talking about like DNA is the genetic material, no? But what is said is, previously when RNA is, means DNA seems to be evolved from RNA. Okay. So what is said that previously in the ancient time of ancient world of life, the RNA would have been the initial genetic material. From that, the cells would have acquired different modifications to have the DNA as the genetic material. So that concept is, in short, I told you there is a elaborate explanation for that. Okay. So this is a brief idea about your RNA. Anything else anyone want to share? No. RNA can have stem loop kind of structure because it is single stranded, no? So to protect itself and to have more stability, stem loop means this sort of structure. This is paired one. See, this is the paired one that is called as stem, and this is unpaired thing that is called as loop. So sometimes to enhance the stability, RNA forms the stem loop kind of structure. Okay. Then there is a famous technique to quantify the RNA that is called as RT-PCR or real-time PCR. Okay, just know the name. There is no need to go for the detail for you right now. So there you can means as the protein, as the RNA is getting synthesized, you can see this amount of RNA on screen directly. Okay, then okay, that's it. That was the content I wanted to discuss. I hope you got an idea. Okay. So I thought like whenever this is not part of your regular class, this is just for your what means refreshment of your what you have read and what you have studied. If we discuss, you may get some better idea. That's why I thought of this. What do you think about this class? Whether we can continue some discussions like this, or yeah, please
Yes,
give me
ma'am.
Continue,
your
ma'am.
feedback.
Please. Continue, ma'am. Continue, ma'am.
What about others?
Yes, continue, ma'am.
Okay. So any further changes you want in this sort of discussion? Means any modification you want to make? Okay, if more people will be there, means that will be good. I know it is winter and it is morning. Why I chose morning is I can wake you up at least, and in morning we can have some product discussion. So that will what just brush up your mind, and you can kickstart your day. Okay, then. So this is all about today's discussion, and whenever I'll find time, I'll discuss some other topics also. Thank you for joining. If you have any query, what we discussed today, you can ask. I think you people are half sleep, is it? Till you broke your nice good morning sleep and joined the class, that is good. Okay, then continue. Good morning, wake up and study, do your work. Your day is waiting for you. Bye bye.